For decades, one of the most glaring blind spots in genetic research has been the absence of Arab populations from large-scale genomic studies. Although Arabs make up nearly 6% of the world's population, they are largely absent from genomic research datasets, limiting understanding of disease patterns in Arab populations and hindering effective prevention, diagnosis, and treatment. This underrepresentation meant that the genetic architecture, population history, disease patterns, and evolutionary story of approximately 480 million Arab people across the Middle East and North Africa remained largely unmapped. Now, in 2025 and 2026, this situation is rapidly changing. Researchers in Qatar have unveiled a high-resolution map of the genetic structure of Arab and Middle Eastern populations, providing new insights into human history in the region and ancestral patterns that may help to explain local human traits and disease risks. Combined with major initiatives like the first Arab pangenome and extensive studies across multiple regions, scientists have begun assembling a comprehensive genetic picture of Arab populations, and what they've discovered has surprised even experts in the field. The patterns emerging from this research reveal a complexity and diversity that challenges previous assumptions about Arab genetic identity, disease susceptibility, and migration across the ancient world. The findings have profound implications, not just for understanding Arab populations themselves, but for precision medicine, disease prevention, pharmaceutical development, and our broader understanding of human genetic diversity. The research gap. Why Arab populations were invisible in genomics. Understanding the significance of current research requires first understanding why Arab populations have been so understudied. The answer lies partly in historical accident, partly in scientific infrastructure, and partly in geopolitical factors. Most large-scale genomic studies in the 20th and early 21st centuries focused on populations that were easiest to study, primarily European and East Asian populations in wealthy, research-intensive countries. This is the first genetic anthropology study on Arabs in MENA, Middle East, and North Africa region, with previous meta-analysis, including 100 populations from 36 Arab and non-Arab communities, comprising 16,006 individuals, evaluating the genetic profile of Arabs using HLA class or Y and class II genes. While some research had occurred, it was fragmented, limited in scope, and often focused on specific health conditions rather than broad population genetics. The lack of Arab representation in genomic databases creates a cascade of problems. Reference genomes, the baseline against which genetic variation is measured, were built primarily from European ancestry samples. This means that genetic variations common in Arab populations but rare or absent in Europeans were often misidentified as rare diseases when they are actually population-specific variants with no disease implication. Conversely, disease-causing variants that are relatively common in Arab populations, but rare in European samples, might be missed in genetic screening protocols. For pharmaceutical development, the bias toward European genetic reference data has meant that drug efficacy and safety data are toward European genetic backgrounds. A drug might be approved as safe based on studies in primarily European populations, but cause unexpected side effects in Arab patients whose genetic background processes the medication differently. This gap in research created not just a scientific problem, but an equity problem. Arab populations deserve the same level of understanding about their own genetics, health, disease patterns, and evolutionary history as any other population. The recent research initiatives represent a correction of this historical imbalance. Heirs of admixture, the mixing of different ancestral populations over thousands of years. These ancestries contributed significantly to European, South Asian, as well as South American populations, likely as a result of Islamic expansion over the past 1,400 years. In other words, not only have Arab populations been shaped by incoming migrations, but Arab ancestry has spread outward to other regions, particularly during the Islamic period. This discovery fundamentally challenges the notion of static, unchanging populations. The genetic history of Arab peoples is one of continuous movement, mixing, and adaptation. Arab identity, which today is primarily linguistic and cultural, was shaped by thousands of years of biological and cultural mixing. Disease patterns and medical significance, understanding Arab-specific genetic risk. One of the most surprising and medically significant findings concerns disease patterns unique to Arab populations. 1. Arab populations have a unique genetic heritage shaped by long-standing traditions, including frequent first and second cousin marriages, which has contributed to a high prevalence of rare diseases in the region. This discovery was shocking to many because it reveals how genetic patterns, both beneficial and harmful, have been shaped by cultural practices. Consanguinity, or marriage, between related individuals is common in some Arab communities, for both cultural and historical reasons. While consanguinity can strengthen certain social bonds and preserve disease-causing variant increases dramatically, making rare genetic diseases more common in consanguineous populations. The research has identified specific disease patterns in Arab populations that require targeted prevention and screening strategies. Identifying the genes associated with leukemia in the Arab population is crucial for effective preventive and treatment strategies, with genetic associations playing a probable role in the 2. Eastern Mediterranean region, which includes the Arabic countries, ranking highest for age-standardized mortality rate. For leukemia, specifically, the Arab Eastern Mediterranean region shows particularly high mortality rates, suggesting that both genetic and environmental factors may be contributing. Understanding the genetic underpinnings of this disease pattern could lead to earlier detection, better treatment strategies, and ultimately improved outcomes for Arab patients. This finding extends beyond a single disease. The genetic research has identified numerous conditions that appear at elevated frequency in Arab populations, including specific forms of inherited blindness, rare metabolic disorders, certain forms of kidney disease, specific hearing loss variants, various neurological conditions. The key insight is that these disease patterns cannot be predicted from European genetic data. A genetic counselor using European reference data might miss variants that are disease-causing in Arab ancestry individuals. Precision medicine, the paradigm of tailoring medical treatment to individual genetic backgrounds, requires that each population understands its own genetic health profile. The pangenome revolution, moving beyond single reference sequences. One of the most significant recent breakthroughs has been the assembly of the first Arab pangenome, a development that represents a fundamental shift in how genetic variation is understood and represented. Traditional genetic research used a single reference genome, a sequence representing one human genome, used as the baseline against which all genetic variation in other individuals is measured. This approach has a major limitation. It obscures the normal variation present in human populations. Variants that are common in one population but absent from the reference genome can be misidentified as rare or pathogenic, when they are actually normal population variation. A pangenome, by contrast, is a collection of many genomes that together capture the genetic variation present in a population. A study published in Nature on the assembly of the first Arab human pangenome marks a major step toward more inclusive genomic research. This pangenome, by capturing the variation present in Arab individuals, provides a fundamentally more accurate reference for understanding Arab genetic diversity. The implications of this pangenome are profound. When genetic counselors, researchers, and clinicians use the Arab pangenome as a reference, they can distinguish between normal variation common in Arab populations, rare variants with uncertain significance, variants that are truly disease-causing. This distinction is essential for precision medicine. A genetic variant might be rare in European reference data, but common in Arab populations. Using the Arab pangenome, this variant would be correctly identified as normal variation rather than as a pathogenic rare variant. The development of population-specific pangenomes for multiple populations, not just Arab populations, but African, South Asian, and other underrepresented groups, represents a correction of long-standing biases in genomic research. It is a shift toward a truly global, inclusive understanding of human genetic variation, genetic markers and ancestry patterns, understanding individual identity. The research has also revealed specific genetic markers that characterize Arab populations and allow researchers to trace individual ancestry with unprecedented precision. The most frequent alleles in Arabs are A01, A02, B35, B51, DRB103-01, DRB107-01, DQB102-01, and DQB103-01, while DRB103-01, DQB102-01, and DRB107-01, DQB102 are the most frequent class II haplotypes. These HLA, human leukocyte antigen markers, which are part of the immune system, show patterns in Arab populations that are distinct from other global populations. Additionally, Y chromosome and mitochondrial DNA studies have revealed haplogroup patterns distinctive to Arab populations. The most common haplogroups in the Middle Eastern populations were J1A, 29.4%, while in the African populations, E1B1A, 43.2%, was the most prevalent. The J1A haplogroup is particularly common in Arab populations and reflects paternal ancestry patterns dating back thousands of years. The revolution you can hold in your hands. Imagine holding a tiny vial of saliva in your hands and knowing, with scientific certainty, that your great-great-great-great-great-great-great-grandfather walked across the frozen Beringia land bridge 15,000 years ago, chasing herds of woolly mammoth. Or that your ancestors survived the Mongol invasions of Central Asia, traveling Silk Roads that no Western maps documented, or that your bloodline carries the genetic stamp of ancient mariners who sailed across the Indian Ocean when Europe was still locked in ice ages. This isn't science fiction anymore. This is 2028. This is real. This is happening right now. The year 2028 marks a watershed moment in human history, not because of political events or technological breakthroughs in artificial intelligence, but because for the first time ever, billions of ordinary people can access the secrets hidden in their own DNA. A saliva sample, a lab test, a few weeks of waiting. Then, boom, your entire ancestral narrative unfolds before your eyes like an ancient map suddenly becoming visible under ultraviolet light. Welcome to the era when your DNA became your personal historian. From mystery to certainty, the DNA revolution explained. Let's rewind to understand how we got here. Just 30 years ago, in the 1990s, genetic ancestry testing was the exclusive domain of academic researchers with fancy lab coats and grant funding. It was exotic, expensive, and frankly, kind of unreliable. Then came the 2000s. The Human Genome Project wrapped up, suddenly flooding the scientific world with genetic data. Researchers started mapping where genetic variations appeared across different populations. A scientist named Spencer Wells launched the Genographic Project, turning the study of human migration into something almost adventurous, tracing the epic exodus of humans out of Africa across every continent on Earth. But here's the thing, those early tests were like looking at the world through a keyhole. Mitochondrial DNA testing could trace your mother's line, going back thousands of years. Y chromosome testing did the same for fathers. But what about everyone else in your family tree? What about the intricate tapestry of heritage that comes from all your ancestors, not just your maternal grandmother or paternal grandfather? Enter the 2010s and 2020s, the era of whole genome sequencing and autosomal DNA analysis. Suddenly, scientists could examine DNA across all 23 chromosomes. Simultaneously, not just the scraps and remnants of ancient maternal and paternal lineages, they could detect genetic admixture, evidence that your ancestors had mingled with other populations. They could identify rare genetic variants that pointed to specific ancestral populations. They could build algorithms so sophisticated that your genetic code could be compared against reference databases containing the DNA of millions of people worldwide. By 2028, the technology had reached a level of sophistication that would have seemed like pure magic just a decade earlier. What you actually get, your personal ancestral odyssey. When you submit your DNA in 2028, you're not getting a simple pie chart saying 50% Irish, 30% Italian, 20% unknown. That's child's play. That's what your great uncle probably did back in 2015 and thought was cutting edge. No, in 2028, you get something infinitely richer and more thrilling. The ancestral map. Your results come back with a detailed, color-coded map showing where your ancestors were located during different time periods, not just East Asia, but 14% from the Yangtze River Valley during the Bronze Age, around 1500 BCE, when your ancestors were among the first to cultivate rice in organized agricultural settlements, or 8% from Mongolic populations of the Eurasian steppes, roughly 1200-1400 CE, when your ancestors were riding horses across vast grasslands, possibly even involved in the expansions initiated by Genghis Khan. The specificity is staggering. The precision is almost spiritual. Migration timing. Your report includes when your ancestors moved from place to place. Genetic mutations spread in waves across populations. Researchers have mapped these waves with remarkable accuracy. So when you see 11% ancestry from sub-Saharan Africa with migration to North Africa dated 300-500 years ago, that's not a guess. That's based on the documented spread of specific genetic markers during that historical period. Ancient DNA connections. Here's where it gets genuinely weird and wonderful. Many people carry genetic traces of ancient, sophisticated Bronze Age culture that rivaled ancient Egypt and Mesopotamia in complexity, or from ancient Egyptian populations, or from the mysterious sea peoples who roamed the Mediterranean 3,000 years ago, their origins still debated by historians. Most large-scale genomic studies in the 20th and early 21st centuries focused on populations that were easiest to study, primarily European and East Asian populations in wealthy, research-intensive countries. This is the first genetic anthropology study on Arabs in MENA, Middle East, and North Africa region, with previous meta-analysis, including 100 populations from 36 Arab and non-Arab communities, comprising 16,006 individuals, evaluating the genetic profile of Arabs using HLA class or Y and class II genes. While some research had occurred, it was fragmented, limited in scope, and often focused on specific health conditions rather than broad population genetics. The lack of Arab representation in genomic databases creates a cascade of problems. Reference genomes, the baseline against which genetic variation is measured, were built primarily from European ancestry samples. This means that genetic variations common in Arab populations but rare or absent in Europeans were often misidentified as rare diseases when they are actually population-specific variants with no disease implication. Conversely, Disease-causing variants that are relatively common in Arab populations, but rare in European samples, might be missed in genetic screening protocols. For pharmaceutical development, the bias toward European genetic reference data has meant that drug efficacy and safety data are toward European genetic backgrounds. A drug might be approved as safe based on studies in primarily European populations, but cause unexpected side effects in Arab patients whose genetic background processes the medication differently. This gap in research created not just a scientific problem, but an equity problem. Arab populations deserve the same level of understanding about their own genetics, health, disease patterns, and evolutionary history as any other population. The recent research initiatives represent a correction of this historical imbalance.